Hey everyone, welcome to Professor Long's lectures in microbiology. As you guys know, these videos are intended for use by students who are enrolled in my class. If anyone else out there in YouTube land finds them helpful, by all means use them. If you do like my videos, please hit like and or subscribe. It gives me some feedback so that I know whether I should continue to do these. These are all very roughly done in a single take. So in the future I plan to update these and make them a little bit more smooth and professionally done. But I just don't have the time right now. Now, also know that if your instructor goes over greater detail or more detail or in other areas than I do, by all means learn the information that your instructor wants you to know. I have a different way of teaching oftentimes where I start with the simple stuff and then later on we're going to add more detail. So I like to keep it simple early on. Otherwise, a student can become overwhelmed with just volumes of information and it's very difficult to keep track of it all. Now, what we've been talking about is the differences between eukaryotes and prokaryotes. Cells that have a membrane-bound nucleus and membranous organelles, and cells that don't, the prokaryotic cells. And prokaryotic cells encompass the two domains, archaea and bacteria. And because this is a health science micro course, we're really going to focus on bacteria, which cause a lot of the illnesses and have the biggest impact on health and disease. And so, when we talk about bacteria, in the previous video, not only did we compare and contrast eukaryotes and prokaryotes, and we listed a number of characteristics which you should have in your brain memorized already, and be able to draw the cells and talk about the membranous organelles, what are the functions of those organelles, and which organelles do we have in prokaryotes. And we also talked about some extracellular features. One of the things that we talked about was things like called fimbriae and pili. We're going to reiterate those a little bit today. Flagella, we're going to talk a little bit more about those. We're also going to talk about the shapes of bacteria. Bacteria come in different shapes depending on which genus and species that we're talking about. Now, when we talk about bacterial shapes, we're talking about morphology. Morph or morpho refers to shape, so morphology is the study of the shape of something. And bacterial morphology would be the study of the shapes of bacteria. If I say that a type of bacterium is monomorphic, that means it has one shape. And essentially what that means is all the bacteria within this genus or species will all have the same shape. If one Bacillus subtilis is bacillus in shaped, then all Bacillus subtilis will have that if it's monomorphic. Okay. If we talk about what's called pliomorphic, then that means many shapes. There are a few bacteria. Most are monomorphic, meaning if it's a particular genus and species, then everything in that genus and species will have the same shape. If it's pliomorphic, then within that species, they can have different shapes. And we'll talk about that in some detail. So when I talk about the shapes of bacteria, there's a number of shapes that bacteria can come in. Okay? One is called coccus. Coccus is singular, or we can say cocci, or some say cocci. This would be plural for the word coccus. So in Latin, you wouldn't make this coccus. It's coccus or cocci. And these tend to be spherical or round in shape. Okay? If I talk about something being a bacillus, or again, plural would be bacilli, then what we're talking about things that are rod, or what I like to describe as cigar-shaped. Okay? And then we can also talk about those that are spiral-shaped. And I'm not going to write spiral-shaped because you know what spiral means. But there's different types of spiral-shaped bacteria. One group is called Vibrio. Vibrio are almost like a comma shape. They are spiral, meaning that they have structures underneath their membrane that allow them to be spiral and sort of corkscrew their way through things. But those proteins are structured to where they end up having a bend in them and looking somewhat spiral, somewhat comma shaped. There's another one called Spirilla. Spirilla really are sort of spiral in shape. And there's another group called Spirochetes. And spirochetes tend to be really long and spiral in shape, okay? So, when we talk about these different bacteria, I want you to know, and I put spirochetes. There's only one T in spirochete. Okay? So, I want you to understand when I use these terms, cocci and bacillus and spiral in shape, what I'm talking about. And later on, we're going to learn the names of some specific bacteria that fall under each one of these categories. Now, when it comes not only to the shape, we can also talk about the arrangements of these bacteria. Okay. Oh, by the way, I knew I was leaving one out. There's one called coccobacillus. And coccobacillus is sort of something between being perfectly round, like this one, or being rod-shaped. These are more oval-shaped. And when I say oval, they're kind of tapered at the ends. They're kind of like somewhat round, like someone took a bacillus and started to smash it, but they're not quite the same shape as a little cigar. So we can have cocci, we can have bacilli, and then we can have coccobacillus, which would all be different. Cocci, bacillus, coccobacillus, kind of a fusion of the two. Now, once we understand what the shapes are, one of the things that we can talk about is the arrangement of these bacteria within their shapes. So when I talk about arrangements of bacteria, I'm talking about how do they group together, okay? So anytime we use the term diplo, di means two, so diplo means that they are in pairs. For example, if I tell you there's a bacterium that is a diplococcus, then what that means is there's two spheres stuck together, and all over the microscope I'll find them in pairs. If I tell you that one is diplobacillus, since bacillus means rod-shaped, they'll look like two little cigars associated with each other all over your slide. Okay? Now, if I use the term strepto refers to a chain of these guys. And so if I talk about something that is streptococcus, what that means is, excuse me, sorry about that, but I'm not going to start this video over. I can have a whole bunch of these cocci-shaped bacteria in a long chain. If I were going to talk about streptobacillus, then I would have a whole bunch of these rod-shaped bacteria all connected to each other in long chains. That's all that strepto means, okay? And by the way, sometimes the name of the bacterium is based on this. Like there's one called Streptococcus pneumoniae, okay? Or Streptococcus pneumoniae. So the name tells you what you're looking at. Now, I'm going to erase this drawing because, well, let me see if I can include all this in the space that I have. When I talk about staphylo, what this means is a group or cluster. And I think of a cluster of grapes. So if I have a whole bunch of bacteria that are clustered together like this, and they're all round shaped, then I would call this a staphylococcus bacterium. Like Staphylococcus aureus means they're clusters of little cocci-shaped bacteria. If I had a whole bunch of rod-shaped bacteria, then these would be Staphylobacillus. So the name tells you something about the arrangement of them. Some are just singular. So we might just have a cocci-shaped bacterium, but if they're in pairs, we call them Diplococcus. If they're pairs of rod-shaped, we'd say Diplobacillus. We can have Streptococcus and Streptobacillus. We can have Staphylococcus and Staphylobacillus. And those are just descriptions. Now, there's a few other ways that we can describe them. I'm running out of room here, so I'm going to erase all of this. And I want to include some of these names. These are a little bit more rare, and you won't see them as often, but they are somewhat, they do fall under the arrangements or categories of bacteria. So, one of them is a tetrad. Tetra means four. So when we say a tetrad formation, then we mean 4 of them stuck together in a little group. And then there's some called sarcinae, and sarcinae is a group of 8 in a cube sort of shape. So I would have 1, 2, 3, 4, and then I can have another one behind each one of these, and they're sort of in a little cube, like if you took 4 little balls and put them together, okay? Or actually 8. So I've got one behind the other, and that's really not a very good drawing, but I would have them like this, and then another one stacked behind each one of these, okay? We would call those sarcinae in shape. Now, when I talk about the spiral bacteria, by the way, one of the things I want to step back and talk about is that in the spiral bacteria, they have what we call axial filaments. And I mentioned this before in a previous video, but basically what happens is I can have the cell wall here of these guys. Outside the cell wall will be the axial filaments, which are some proteins in here that kind of wrap around. And then outside the axial filament, there is somewhat of a sheath that covers them that allows these filaments to cause the bulges in the outside. And essentially what happens is the axial filaments can wrap around the bacterium, and as the bacterium moves it, it allows the bacterium to spiral almost like a corkscrew and kind of screw their way through fluids and things. So we refer to these as the axial filaments, and that's what's found in the spirochetes and the spirilla and in the vibrios, the comma-shaped ones, okay? Now, another structure that I want to talk about when it comes to these bacteria is this, and we mentioned this before. Let's say I take another rod-shaped bacterium, and on this bacterium, I can have these structures that stick off. And if they're rather, short structures, and not these long structures called flagella. Flagella are long whip-like tails that are made out of protein that can whip around and allow for what we call motility, allow them to swim around. Some of these other structures that are shorter can be called a fimbria, or fimbriae for plural. And fimbriae are smaller hair-like structures that allow the bacteria to attach to other cells and sometimes cause some problems. Now, I also mentioned this before. Some bacteria can have little structures sticking off called pili, and that will allow them to attach to other cells. Pili are rather short, hair-like structures, and if one bacterium attaches to another with its pilus and it can inject its DNA into the other, we call this process conjugation. Conjugation would be when one bacterium uses its pilus, called a sex pilus, to stick to another bacterium and then move a plasmid over or some genetic material and altering the genetic makeup of this guy. And again, a plasmid is a piece of extra chromosomal DNA that can be incorporated into the chromosome if necessary so that we can run those genes and turn them into proteins. And when we don't need it, we can pull it back out. And some of these plasmids actually allow some bacteria to be able to break down antibiotics. And again, we're going to talk about that later in the semester. So conjugation is an important concept. Think about a conjugal visit in prison where people actually get to hook up. Well, it's where two bacteria hook up together with a sex pili, and one can transfer genetic information. So we've talked a little bit about fimbria and pili in some detail before. I just wanted to mention it again. Now, I want to focus on flagella for a minute. And when we categorize flagella, the flagella in bacteria, then we can say whether a type of bacterium is atrichous, which means it has no flagellum. Bacteria that lack flagella are said to be atrichous. Okay? If I say that a bacterium is peritrichous, well, you know peri means around from perimeter. That means that the flagellum are all around it. So I could have one bacterium that's rod-shaped have no flagella. So we would say it's an atrichous bacterium. If I have one that looks like this and it has flagella all over it, then we would say that one's peritrichous, okay? Now, a couple of other terms that we can use is we can say whether the, we can talk about whether we have what's called flagellar polarity, whether they are polar or not, like the poles of the earth. So, if I talk about something being monopolar, again, mono means one. If I have a bacterium that has flagella at one end, then I would say that it's monopolar bacterium. Okay? Or I could also say that if, well, I'm going to get to these terms in a moment. This would actually fall under a category that we call lophotrichous. Okay? Lophotrichous means they have a tuft of flagella at one end. Okay? So, now I could say that this is lophotrichous and monopolar because it has a tuft. Lophotrichous usually has a tuft of flagella, a small group of them, all sort of anchored in the same area. So lophotrichous and monopolar would mean I have a group of them at one end, and usually they are that way. Now, a couple of other terms that we could use or we could talk about is we could say if something is amphitrichous or monotrichous. So I'm going to erase this picture right here, and I'm going to talk about this. Okay? If I say something is amphitrichous, amphi means both, and trichous refers to the flagellum. So an amphitrichous bacterium could have a flagellum at both ends. And if it's monotrichous, monotrichous means that it has a single flagellum. So some bacteria are polar. They exhibit flagellar polarity, meaning there's a flagellum at one end, at both ends, or a group of flagella at one end. If it's a group of flagella, a tuft of them, we call them lophotrichous. If it's at both ends, it's amphitrichous. And if they're monotrichous, they have a single flagellum. If they're peritrichous, they have flagella all the way around them. And if they're atrichous, they have no flagellum. Okay? So we'll use this terminology sometime when describing specific bacteria. Again, right now, I don't care if you know which ones are which. If I start throwing out all these scientific names at you amongst the vocabulary you're already learning, I believe that a lot of students just kind of get flustered. I know I saw it with my classmates. I had the ability to just sit and memorize and force myself to do so, but a lot of my classmates just like, man, this is too much. I got to take another instructor or something. And they switched when I was a student in microbiology. Anyway, so right now I want to reiterate this. We've talked about the shapes of bacteria. You should know the main shapes. Coccus means round or spherical. Bacillus means rod-shaped or cigar-shaped. And Vibrio would be comma-shaped, and they belong to a class of spiral bacteria that include Vibrios, which have a axial filament that makes them spiral, but it's such that they are bent. You have Spirilla, which are spiral-shaped, and you have Spirochetes, which is a whole class of them that are also spiral-shaped. That axial filament is outside the cell wall, but underneath a sort of sheath of structures that allows them to corkscrew and move. We're talking about motility of bacteria, and some bacteria are extremely motile, meaning they can move, and all the spiral bacteria move through corkscrewing through fluids and things. Other ones are called flagellates. Not all bacteria have flagella, but if they do, then they can whip those little flagella and swim through fluids. So we talk about them being trichous. Monotrichous has a single flagellum. Atrichous has no flagella. And if we talk about them being peritrichous, they have flagella all the way around. If they are lophotrichous, they have a tuft of them. And amphitrichous means they have a flagellum at either end. You should know what that term means, and that's the kind of information I'm going to expect you to know. Now, one of the last things that I want to talk about is I want to talk about the structure of the flagellum. And if you see me stepping off screen, I do that on purpose at times if I need to take a drink of my coffee or some hot tea to clear my throat. Now, if we were to look at a bacterium that has a flagellum sticking off of it, or more, we're going to look at a single flagellum, but I'll draw this one with a bunch. If I start to cut into the covering here, you guys should know that the layers of the bacterium would be that they can have a membrane here that is a phospholipid bilayer. It's very similar to the membrane of eukaryotes, but again, it lacks compounds called sterols. And then outside of that, we can have a thicker layer that we can call the cell wall. And depending on if they're gram-positive or gram-negative, the cell wall can vary, but the cell wall will be made of some peptidoglycans. It can be a very thick layer of peptidoglycans, as in gram-positive bacteria, or it can be a thin layer of peptidoglycans with an outer membrane out here, as in gram-negative. So this would be one of our gram-positive bacteria for now. And we were simply just cutting into this back end here. And one of the things that we can see is that the way that the flagellum is attached is the flagellum has a piece that sits, a protein-like structure that is very often somewhat bent, called the hook. And the function of the hook is that it attaches the flagellum, or actually what we call the filament of the flagellum, to what we call the basal body. The basal body is a structure that is anchored to the cell. Oops, I should use a different color so that you guys can see this. Okay, I'll use black for the basal body. Okay, so the basal body attaches the filament to this structure here, and the basal body is what is anchored to the cell wall and the cell membrane. Okay, now attached to this will be a long whip-like structure, and I'm not going to draw the whole thing. It's usually uniform in thickness, but this part is called the filament. The filament is a long string of proteins that have the ability to move next to each other so that we can whip the flagellum around and allow the bacterium to swim. The filament is attached to the cell wall and cell membrane by a hook, and what's attaching it here is the basal body. And then inside the cell, there are some structures that we can call the motor. And it's not a motor like in your car, but it is a group of proteins that allow movement between them, that allow the entire filament to be moved so that the bacterium can swim around. Okay? Now, one of the important things that I want you guys to know is that the basal body can be different between gram-negative and gram-positive bacteria. Okay, in gram-positive bacteria, there's really only what we call the inner ring. So I really didn't draw this accurately, but attached to the hook and the basal body, there's a ring in here that attaches it to the inner membrane. And so in gram-positive bacteria, they only have a single ring. If I were to draw a gram-negative bacteria, so this one would be gram-positive. In a gram-negative bacterium, we have these two lipid bilayers, and I'm not going to draw all the lipids. I guess I will. And we'll have a motor protein in here. And then we'll have our cell wall, which is a thin layer of proteoglycans And then we'll have the outer membrane. And again, these are phospholipids. You should understand the structure of phospholipids from both the chemistry lecture or from your previous biology classes, especially if you had me for A&P. Now, when it comes to the basal body will have two rings, an outer ring and an inner ring for the basal body, and then we would have the hook, and then you would have the filament sticking off of this. The filament is the part that whips around and provides the motility, so to speak. The hook attaches the filament to the basal body. The basal body is what anchors the entire structure to the cell wall and the cell membrane. And on gram-positive bacteria, they only have a single ring on the inside. On gram-negative bacteria, they have two rings to their basal body, and that's one of the distinguishing characteristics. The motor protein is what moves the flagellum for the motility, okay? And that's about as simple as, or as complex as I want to get. I want to keep it real simple right now. We could spend weeks talking about the different structures and the actual proteins that make up these structures and how they're assembled and come together. But that's for a higher level microbiology class. Now, let me browse through my notes real quick, make sure there's nothing else I want to cover that I haven't already covered. I just want to make sure I don't leave anything out. No, that should be it. All right. So anyway, in these two videos that we've covered back to back, I hope that you guys learned about the differences between prokaryotes and eukaryotes and can list those characteristics. Those are the things you're going to be asked on exams. Which one is has a membrane-bound nucleus, which one does not. What are the types of ribosomes found in prokaryotes or eukaryotes? Prokaryotes have smaller ribosomes, 70S subunits, or 70S or 70 Svedberg units, where eukaryotes have 80S ribosomes or 80 Svedberg units. And again, you don't need to understand fully what Svedberg units are, you can look them up. And a number of the other differences that we talked about, whether they have a cell wall or not. Some eukaryotes do, most prokaryotes do, most bacteria have a cell wall. And then again, you should know the difference between the gram-negative and the gram-positive. The gram-negative have a very thick proteoglycan layer. Gram-negative have a very small one. Gram-positive thick proteoglycan, gram-negative. I always think of the positive as the plus proteoglycans. Extra proteoglycans are added into the cell wall. Gram-negative are missing or lacking some of those proteoglycans. Now, one of the differences, gram-negative also have an outer membrane. And then when it comes to the shapes of bacteria, you guys, well, you should also know about the capsule. You should know about fimbria and pili and what their functions are. You should also know what the flagellum does. It provides motility. The pili and fimbria provide attachment to other cells. And the sex pili allows for conjugation. We've reiterated that a number of times. You should understand the different shapes of bacteria from coccus to bacillus to the spiral. And under spiral, you should know Vibrio. You should know the spirilla and spirochetes. And you should know about the axial filaments, providing that spiral shape to allow them to corkscrew through things. That's how they move. Flagella provide motility to bacteria that are flagellated or have one. If they are atrichous, they lack a flagellum. And you should know the different terms associated with that. Monotrichous, having one. Peritrichous, all the way around. If they exhibit polarity, then the flagella is only on one side or the two ends. If it's amphitrichous, it has a flagellum at either end. If it's monotrichous, it has a single flagellum at one end. If it is lophotrichous, it has a tuft of flagella at one end. Now, when it comes to the structure of the flagellum, I want you to understand that the motor protein provides, that is what moves the filament. As the filament moves, it provides the motility. The hook is what attaches the filament to the basal body. The basal body is what attaches the whole thing to the cell wall and cell membrane. Gram-positive bacteria have a single inner ring to their basal body, and gram-negative have a ring for the outer membrane and the inner membrane. All right? I think that's simple enough to follow. You guys need to make note of that, and those are the things you need to be studying for now. When we get past the first exam, usually the first exam in a micro class encompasses this information. Once we get past that, we will move on to some more complex views of all of these things. But we certainly have covered a lot. You guys should understand the history of microbiology, why microbiology is important in our lives. You should understand some basic chemistry. You should understand a little bit about the microscope, and you should be able to answer a lot of questions about bacteria and prokaryotes and eukaryotes. All right? So I hope you guys had some fun. I hope you had as much fun as I did. I hope you learned something. I hope this was informative. And if it is, again, please hit like and subscribe. That helps me out. And I'll see you on the flip side of the next video. Thanks for watching.